That'll move us on to our fourth and final speaker for the session, Dr. Tim Sweeney. Tim is a physician and data scientist with over 15 years of research experience in sepsis. After completing his MD PhD, he was a surgery resident for 4 years and then reformed and became frustrated with the current diagnostic tools for infection, went on to do postdoctoral studies in biomedical informatics, and worked on ways to identify new ways to diagnose infections based on sort of, quote, reading the immune system. In 2016, he founded Inflammatix to commercialize this research and has served as the company's CEO. The company has 24 clinical studies currently enrolling and has been supported by several public and private partnerships. So thanks, Tim, and it's all yours.
Great. Thanks for the introduction. So I was asked to speak today a little bit about some of the challenges that we face in building a novel diagnostic for sepsis. And definitely let's see if this doesn't work. Oh, sorry, there's just a lag. I apologize. Go back to disclosures. Oops, user error. Maybe if we can go back to the disclosures slide here. The lag is killing me. I have no idea what I'm doing. We'll do that. So I am an employee and stockholder in Inflammatix. I will very specifically not talk about the products that we're building here, but sort of general challenges that we face in the space, many of which concern the challenges that we have in defining sepsis. So you've heard many times today the sepsis 3 diagnosis dysfunction caused by dysregulated host response to infection. As a physician, I think this really breaks down into two axes, which is there an infection present, and is there life-threatening organ dysfunction present? How sick is the patient? And the problem in creating novel diagnostics is that we have to come up with some kind of solid definition for what each axis represents. We have to know who has an infection. We have to know who has organ dysfunction. One can imagine that if you're trying to define organ dysfunction and the definitions keep changing, it's a rather hard thing to do. This was a study published by Manu Shankar-Hari a couple of years ago, showing the difference between sepsis 2 septic shock and sepsis 3 septic shock. Some patients have one, some patients have another, some patients have both. Which definition of organ dysfunction is quote unquote correct for us in the sepsis diagnostics community to target? When it comes to defining who has an infection, you heard a little bit earlier from Steve Opal about some of the benefits that are coming to bacteremia diagnosis. But we also know that the diagnosis of bacteremia is actually a minority of patients with sepsis. Most patients with sepsis have a severe non-bloodstream infection. And the reality is that we don't necessarily just want diagnostics for patients with sepsis. We want diagnostics for patients that are suspected of sepsis, some of whom may not have an infection, some of whom may have an infection that's not coupled with life-threatening organ dysfunction. And so there's really a critical need for novel diagnostics for the presence of an infection across the whole spectrum. The issue with blood culture and some of its sensitivities are well known. There are some people in the space working on next-gen sequencing solutions. But the interesting thing that we found in general is that pushing the bounds with NGS is only a marginal benefit. This was a paper published in Nature Microbiology last year showing that if you sort of follow the black line outside the circle, the things that were found on blood culture on the orange line were also found on NGS by and large. And NGS also picked up some extra potential pathogens, but it picked up a lot of stuff that probably was not pathogenic. It's just microorganisms that are floating in the blood. And you still had a lot of patients that were clinically diagnosed with sepsis that were negative by both methods, meaning that the blood isn't ever going to be sort of 100% sensitive for telling you who does and doesn't need antibiotics. This was, I think, pretty elegantly put together in another study by Jane et al. In the New England Journal in 2015, where 2,300 patients with a radiographically confirmed pneumonia were subjected to a full suite of microbiology testing, bloodstream, blood cultures, nasal respiratory panels, sputum culture, urine serologies, the whole gamut. And across these 2,300 patients where we knew that an infection was present, we could not detect a pathogen in 62% of that cohort. And then if you sort of say, okay, well, let's take a step back. Physicians know who has an infection, right? We can tell. So maybe we don't have the microbiology, but we're pretty good at judging bacterial and viral infections. A great study published in the Journal of Intensive Care last year showed inter-observer agreement of 0.24 to 0.7. I mean, a very, very low inter-observer agreement in physicians looking at a patient's chart and trying to decide whether or not this represents a bacterial or a viral infection. So the reality is it's very difficult to figure out who has an infection, who has organ dysfunction. And then there's a whole different class of sepsis diagnostics, which are these sort of EHR sniffers. I think we'll hear a little bit more about these later. And some other folks have already spoken to some of the EHR-based tools that might use things like labs and vital signs to predict quote-unquote sepsis. But that means you have to denote quote-unquote sepsis somewhere in the patient's chart, right? You know, on the left, this was from the PhysioNet group paper that came out in Critical Care, sort of how to design one of these novel diagnostic technologies. And at some point, you have to say, this patient had sepsis here. This is the time of sepsis. On the right is a 7-year-old paper showing that if you apply different methodologies for defining who has sepsis in a medical record, you can come up with a 5-fold variation in who might qualify. So when you don't have a gold standard, it's really hard to train machine learning appropriately to give actionable information. The final question, of course, is even if you have a useful result, if it doesn't come in the right time in workflow, it doesn't help very much, right? If you had a perfect host response technology that could tell you this patient does or doesn't have a bacterial or viral infection, but it doesn't turn around for 2 or 3 days, no one's going to use it. And vice versa, you know, we do see some ID and AST technologies coming out that certainly improve upon blood culture, shortening things from 5 days to maybe 3 days, but they're not going to change your initial decision on empiric antibiotics, right? It's still a 2 or 3 day late decision. So as a diagnostic test developer, this matters a lot too. Not just can I get the right result, but can I turn it around in an actionable time frame for where the patient is in this really, really complex workflow, an example shown here on screen. And then finally, even if you do build a successful test, meaning that it's technically a success, it has a high sensitivity, high specificity, it can still be a market failure. One of my favorite papers came out of the NICE assessment. This is a UK NHS assessment of the Iridica and light cycle or Septifast tests brought to market by Abbott and Roche. Ultimately, both tests were direct from blood bacteremia detection systems. They were technically adequate, but they were commercial failures. They were, and untold millions were spent bringing them to market and then pulling them from market, because in the end, physicians didn't change their behavior enough when using those tests to show that there was any kind of actual clinical benefit of onboarding them in practice. I think the reason being that most patients who were sick enough to be bacteremic probably were already on broad-spectrum antibiotics, and ID alone without AST probably didn't change those antibiotic choices. But it goes to show that as a test developer, it's not enough simply to be accurate and in workflow. Now you need to be accurate in workflow and doing something that a physician is going to utilize, such that eventually you can gain proper reimbursement and make a real difference in clinical impact. So it's not all bad. The future does hold good things. And I think that I like to think about it really in terms of workflow, right? We have EHR sniffers, of which, you know, in that PhysioNet paper, I think there were 850 some odd applicants. And there are literally now hundreds and hundreds of sort of AI-based sepsis sniffers out there. And the benefit, of course, is that they're instant and they're functionally free for a new patient. But they don't function initially in the emergency department very well, because you don't have a long time recording for the patient when he first shows up. They're very good at risk stratification and prognostication, not very good at identifying who has an infection. And there's always the question of what does a clinician do with data? What do you do if somebody says, you know, shock is impending in 12 or 24 hours? You're not going to pre-press the patient, right? So what is the action that one would identify downstream of this? Certainly biomarker panels are coming, and again, full disclosure, Inflammatix is building one as well. These might function in something like 5 to 30 minutes. They can do the diagnosis of an infection, and they can do prognostication separately. But the key question is going to be, is this marker generalizable? If we have a marker for the presence of a bacterial infection, does it work in inpatients and outpatients? Does it work in medical patients and surgical patients? Does it work in the old and in the young? And that's really going to be a difficult challenge of any sort of attempt to interpret what the immune system is doing. And then finally are going to be the rapid ID and AST. absolutely critical parts of the sepsis diagnostic workflow. Probably even in theory, you're only going to, speed this up to something like 4 to 6 hours, just due to the need for bacteria to grow and then see sort of the phenotypic drop-off. AST is certainly routinely actionable. ID maybe a little less so, and I think we've seen that in the market failures. The good thing is that all of these tests currently have to be used in the concept of a direct from blood infection test has to be used in everyone, most of whom don't actually have bloodstream infections. If they were used downstream of good biomarker panels that could say, yes, this patient likely has a bacterial infection, then the cost wouldn't be so high on this side because you'd only be running them in patients that routinely actually did have a bacterial infection, so they would turn out positive more often. But there's still going to be negative tests that you have to pay for anyway. And then there's this question of genotypic versus phenotypic AST and what's likely to impinge on a physician behavior. And then sort of down here, separately maybe from diagnostics, but clearly on everyone's mind, is some kind of therapy response prediction, right? We heard Monica Shankar-Hari talk about it today. We've heard several of our panelists, Dr. Meyer and Dr. Marshall, talk about it today. Some kind of way to tell not just is there an infection, how sick, but can we use some kind of readout of the patient's trajectory to suggest which therapy is going to be helpful, whether it's which novel immunotherapy or even just what kind of fluid level to strike, which pressor is best. I think that's the promise of the precision medicine sort of next generation in sepsis, and we look forward to seeing it unfold. I think we can move on to the questions now. Thanks very much for the time. I appreciate it.